Okay, so is my screen visible to all of you? Can anyone can tell me, please? So okay, now I'm starting it. So it's already time. So I request all of you to please join on time. And so can anyone tell me, please, that my screen is visible and my audio is audible? Am I audible? Okay, okay. Thank you. So good evening, everyone. I am Simran, a fifth year PhD student at IIT Bombay. So, I am a teaching assistant for this course called Medicinal Chemistry, which is running on NPTEL platform, and the course instructors are Professor Haninath Chakrapani from ISER Pune. So, this is a 12-week course basically, and today this is a week one, which I am conducting right now, the tutorial session. So, we will be meeting every week at 5 p.m. and on Saturday, every Saturday, and this is a 12-week course, so we will be continuing for 12 tutorials. And still since it is a first tutorial, so I'm just focusing on to the concepts, that's the basic concepts of medicinal chemistry. Okay, so the questions are related to the video lectures which already have provided on the NPTEL platform by Professor Harinath Chakrapani. I hope you all have already gone through it. So the people who already have gone through it will know exactly what I'm talking about as the basics of medicinal chemistry. So today we will be discussing some of the questions which are very basic. Like it's not that much basic, like it's a basic. So uh, basically you have to answer those questions in this tutorial and participate well. And whatever your doubt is, whatever you are going through, just tell me, and please make me stop wherever you are not comfortable, wherever you are seeing that you are not getting anything. Please make me stop. I will more than happy to elaborate the things after that. So we'll be starting today's lecture. So, first of all, the questions which comes is, as it is a medicinal chemistry and the drug discovery and drug development is very important. So, the first question is the process of bringing a new pharmaceutical drug to the market once the lead compound has been identified through the process of drug discovery is known as. So, basically, just to focus on the question what it is asking, that first, after identifying the lead compound, how you are bringing a pharmaceutical drug to the market, right? And what is the process called? So, can anyone tell me what is the process among these four options, like drug discovery, drug development, both of them, or none of them? Drug development, very nice. So it is drug development. So what is a drug development? There are actually multiple phases, right? So these are the phases which is here, like first the laboratory test is there, then the early clinical stages, then late clinical stages, and then after that, after having all these processes in hand, you will then basically go into the market for the purchase. So here in the lab, first of all, discovery of the target finding. Basically, you find a target and go through a lead compound, find a lead compound, and then you will go to a preclinical test. What is a preclinical test here? It is the test which you have first done on animals. You can't test any drug directly on humans, right? So first of all you have to try it on animals, right? So for that you can check the toxicity of that particular drug developed, and you can then go through the phase 0, that is the early clinical stage. So here you will now find out that what is the toxicity of the lead compound, what is the limitation, and then you will test it on human. So here the patient range is 10 to 15 humans they will check, and the period is 14 to 18 months this trial has been gone through. So what is this, like they have taken a human and just inject some micro dosages to the human, not the exact dosage they want, the micro dosage, and then they will see what is the effect which is causing, right? Then they will go through the phase 1, phase 2, and phase 3, that is the late clinical stages, and which is performed on various patients. Like the parameter you can see here, that the phase 1 includes the 20 to 100 healthy volunteers. After that, the number of patients and number of volunteers will increase as you go from phase 1 to phase 3. Okay, so this is how you will see how you can see the safety hazard, what is causing actually basically the drug which is acting on the particular target, how it is responding, how your body is responding, how effective that drug is, and how you can test the efficacy. So basically, efficacy, you can say that here you will find out how much dose of that drug particularly be sufficient for your body uh, to basically turn on the target or to attack on that particular target which you want. Okay, so this is how these are the phases of early and clinical, late clinical test, and then it get commercialized into the markets. So this is how a phase of drug development happens. Okay, it take a quite amount of time, right? It is a very tedious and it is a very long process to get a particular drug commercialized. Okay, so this is the phase. I hope everything is cleared till now, which I am discussing. So anyone has any doubt regarding it? Okay, so I'm marking it that you guys are getting my point. So whenever you feel that you are not getting something, please raise your hand. Or if I'm not able to see your hand, just speak it out. Okay. So now we will go through what is the difference between a drug discovery and a drug development. As we have already seen in the table that drug discovery is identifying first stage. This is basically the first stage, that is the identifying the lead compound. So identify the new compound, and then it is solely lab based, it doesn't implies any human or animal test, and then it ends with a lead candidate. So in drug discovery you will end up having a lead candidate in your hand, but you haven't tested it, you haven't done any experiments, just you are focusing on the lab experiments, lab data which you have, and then you will go forward. And when you are going forward, the drug development comes into play, that is evaluates the safety and efficacy of the particular drug, and it is tested on humans, right? And after that, after having so many safety precautions, efficacy tests, and everything possible, then you will end up with having a approved drug. So this is the basic difference between drug discovery and the drug development. Okay, so I hope it is clear, the first question is clear to everyone. So I'm seeing that many of the person are going to join, so I will let them in. Okay, everyone, please join on time. It's requested to everyone. So I hope everyone has already joined now. Okay, yes, one more is coming. Please, guys, don't join now. Okay, so getting back to our questions. So I hope first question is clear to everyone. Now I'm going forward. So now this next question is compound with good activity and selectivity in screening drug discovery is known as. So can anyone answer it? It's a hit compound, lead compound, NDA, IND. The compound which is found to be having a good activity and selectivity in screening during the drug discovery. What that compound is? Can anyone tell me? Any guesses? Lead compound. Very nice. So it is known as lead compound. So please keep in mind that when you are doing the drug discovery, you will have a lead compound in your hand. So lead compound is nothing but a chemical compound that is significant, that shows a significant biological activity against the target which you are setting, right? The potential disease target which you are basically seeing, like that Parkinson disease. So you have a We will talk about it in the previous slide, in the next slide, sorry. So this is the lead compound, which is you are getting after further optimization in the drug discovery. So this is your product of that drug discovery. Okay. So the key characteristic of a lead compound is it should show a moderate to high biological activity. Basically, it should be showing a minimal activity which is very much required for the target, for the mechanism of action of that particular target molecule. Okay. So it also shows a structure activity relationship. What is meant by that? What part of a molecule is responsible for its biological effect and how the structural changes affect the activity. So it should be seeing that whether the lead compound, wherever the lead compound is acting, if it is causing a structural change in the molecule, then how the structural change is then affecting the biological property of that particular thing. So this is how you should have a clear cut picture of that structure activity relationship between your lead compound and the target molecule. Okay. Then the selectivity towards the intended target. Yes, it should be very much selective for the intended target. What it conveys that the lead compound should not be like something which is going here and there in your body, here and there in your proteins. It should be selective for that particular target which you are focusing. Okay. Then it should be acceptable solubility and permeability. Obviously, as we know that there are various ion channels in our body, various enzymes, receptors, which is there. So there should be a acceptable amount of solubility and permeability. What is meant by permeability? Basically, uh, moving throughout the cells. So this is the permeability with that is must for that lead compound. Okay. Then synthetic feasibility. This is the feasibility means that it should be synthetic feasible. Basically, you can prepare that lead compound easily. Okay, it's not like you are doing one thing and then it is not reproducible or something like that. It is not happening. So then the low or manageable toxicity, that is very important. So when you are talking about drugs, you have to manage a toxicity scale. So, how that particular lead compound is toxic to your body? There is a range which is acceptable and that is not acceptable, right? So, that should be on the margin, that should be on the range. So, it is having a low or manageable toxicity. If it is not low toxicity, then it should be a manageable toxicity, means the range of the toxicity level, it should be minimal. Okay. So, these are the key structures for the lead compound. Then, as you have already gone through the lecture videos of the Professor Harinath Chakrapani, so I'm just taking a bit out of there. So he has already talked about the approaches to finding a lead molecule. So how you find out that this is a lead molecule, right? So how you can aim on finding a lead molecule. So it should be a natural ligand or substrate. Now, just think about it that you have already find out, like in textbooks, already there is a compound which is having a manageable toxicity, which is having a higher biological activity, and it is well known, right? And from there, you will develop a compound. So that is known as a lead compound, basically. And from there, you will develop another molecule which is having the higher biological activity or higher potency from that particular thing. So if you take a case of Parkinson disease, so as we know that dopamine higher concentration is not feasible. So feasible, I mean it is not preferable, right? So we know that increasing the dopamine concentration, it is an important aim for treatment of Parkinson disease. But increasing the doses of dopamine doesn't it is not preferable. So that is why now you can see that dopamine is a lead compound, and from there you have to reach a compound which is more active, less toxic, has less adverse effect, and you can talk about that on that. Okay, so therefore dopamine was the lead compound for the discovery of rotibutin, a drug that is used for the treatment of Parkinson disease and restless leg syndrome. So basically, dopamine here is your lead compound to find a new compound which is having a higher biological activity, higher like low toxicity, low adverse effects. Okay, so this is how you will find out a lead compound. So I hope it is clear to everyone. So this is how we can say that a compound which is having a good activity, selectivity in screening of drug discovery is known as lead compound. So is it clear to everyone? Okay, thank you. So then I'm going to the next question, and maybe there is our attendee as well. Okay. Okay. So coming to the question number three, so it is saying that how many people will be selected for phase one trial? The whole market will be under surveillance, 300 to 3000 people, 20 to 300 people, or 20 to 50 people. So based on what I have already showed you, the chart, can anyone tell me if someone has all like seriously looked at it? So how many people will be selected for phase one trial? Okay, that was a close go, but it is 20 to 50 people because we are talking about the phase one trial. Okay. Because in phase 0 you will see that only 10 to 12 or 5 to 10 people are selected, and after that phase 1 you will see that 20 to 50. Okay, nice guess by the way. So this is here. So basically in 1a you will see that 20 to 100 people are there, but not 300. That is very high. Okay, that you will see in phase 2. Okay, or phase 3. So the answer is 20 to 50 people. So now again going to some of this kind of situation, that the next question is human microdosing studies conducted in which clinical phase? Phase 0, phase 1, phase 2 or phase 3? So phase 0, nice, good. So phase 0, because as I have already told you that human microdosing is in phase 0, because before that they will try on animals, and then they will try to inject microdoses to the humans to see how they will react, or they will get affected by that particular microdose. Then they will increase the dose and they will increase the number of people if the clinical test goes right. Okay, so this is again I'm showing it here to just keep in mind that this is the basic phase of drug development, and it is very important to know, and it is very like the handy chart, so that we can see how many peoples, what they are doing, what are the safety measures, what are the time. So you will get everything in one go. Okay. So again someone is joining. Okay. Sorry for the disturbances, guys. So now going to question number this. So here we are going to talk about pharmacokinetics and pharmacodynamics. So I hope you have already studied these terms in the tutorial session, because it is very much clear there, because professor has uh, basically talked about it a bit. So here they are saying which of the following statements best describes the pharmacokinetics. So the statements are the study of how drugs reach their target in the body and how the levels of drug in the blood are affected by various factors. The next point is the study of how drugs can be designed using molecular modeling based on a drug pharmacophore. C point is that a study of how a drug interact with its target binding site at a molecular level. So the next is the study of which functional groups are important in binding a drug to its target binding site and the identification of pharmacophore. So what do you think best describes the pharmacokinetics? Anyone? Option A. Good. So you guys are very clever, huh? You know every answer. Nice. It must be like you have done a thorough study of that particular tutorials. So it's very good. So here the pharmacokinetics means that the study of how drugs reach their target in the body and how the level of drug in the blood are affected by various factors. So to simplify this thing, the pharmacokinetics is the study of kinetics of absorption, distribution, metabolism, and excretion. So basically, it is a ADME. So absorption means how drug is going into the body. So it's basically how it is getting absorbed in the body. Distribution means how the drug is getting distributed all over your organs. And then metabolism is how you are basically converting that drug into small metabolites to get it digested. And then excretion, as you all know, that it is getting removed from the body. So how all of these processes are involved in the um, absorption, distribution, metabolism, and excretion of the drug, and their corresponding pharmacological, therapeutic, or toxic response in mans and animals. So basically, their absorptions, distribution, metabolism, and excretion, including the effect of that particular thing, is known as the pharmacokinetics. Okay. So you have to keep in mind that these four terms are very important when you talk about the pharmacokinetics, that how it is getting absorbed, distribute, metabolized, and excrete from our body. Okay. Then I hope it is clear to everyone what is pharmacokinetics. Okay. So now going to similar term, which is the pharmacodynamics. So the question says, which of the following statement best describe pharmacodynamics? So the option is the study of how drugs reach their target in the body and how the level of drug in the blood are affected by absorption, distribution, metabolism, and excretion. Next is the study of how drugs can be designed using molecular modeling based on a drug's pharmacophore. C point is the study of how a drug interacts with its target binding site at a molecular level. And D is the study of which functional groups are important in binding a drug to its target binding site and identification of a pharmacophore. So, what do you think a pharmacodynamics is among these four? Option A. Okay. So here there is a catch that pharmacokinetics we have learned right till now that it is a process in which the drug molecule is getting absorbed, distribute, metabolize, and excrete from our body. So that is the pharmacokinetics. But that is happening in our body. But how our body is getting response? Like first of all, drug is doing to our body these kind of things, and then how our body is getting affected, how our body is responding to that particular drug. So that is known as pharmacodynamics. Okay, so it is not option A, it is the pharmacokinetics, but option C, that is study of how a drug interact with its target binding site at a molecular level. So, as to simplify these things, I have just taken this one-to-one comparison of this pharmacokinetics and pharmacodynamics. So, as you can see here, pharmacodynamics is a mechanism and effect of medication within the body. How the medication is acting in our body, what it is doing, and how the response of our body is going to happen. So this is known as pharmacodynamics. So the importance is determines the drug efficacy. Basically, as you have taken one of the drug, like if you take paracetamol, right? So if you have fever, you take paracetamol. Then what happened? This medication will goes into your body. It will get absorbed, excrete, whatever it is, and that is a pharmacokinetics. You are taking a drug into your body. Then how it is getting affected, basically how your organs, how your body is getting response, that is a pharmacodynamics. So what happens, you will see that determines drug efficacy. So you know that taking one pill is more than enough to get your to get your treatment, right? How you know that? It is already documented, right? It is already prescribed by doctor. How doctor knows that? So this is how you have done after you have checked, right? You have taken one medicine, you are feeling good, your body is responding good, your everything is fine now. Then you will get to know that this one medication is fine enough, right? You are taking two pills, and then it is going to basically your body is getting responses after the taking two pills. Then you will say that this is the dosage amount you will need to get that particular treatment of the disease whichever you are having. Okay. So here you will determine the drug efficacy. Efficacy means the affinity, basically the power of that drug, basically the power of the drug to treat that particular disease. Okay, so how much amount of that drug is needed for that particular treatment of the disease. So here you will see that you will determine the drug efficacy, that how potent that drug is to your treatment. It helps to select the optimal dose. Optimal means if it is if the disease requires one pill and you are taking two already, so it is not advisable, because that is not an optimal amount. Optimal means just the right amount to get it treated. Okay, so if you are taking one pill, then yes, if your body is very much responding towards the one pill, then it is the optimal amount of that particular medicine. Okay, so pharmacodynamics basically give you a hint of the drug efficacy, help you select the optimal dose, and explain the side effects also. So sometimes you will take some of the medicines and it also got banned in India. Some of the reasons are there because it has severe side effects. So because of that severe side effects, it get banned, it get discontinued from that particular pharmacy. So here this is the side effect which you are getting from pharmacodynamics. Essential for rational drug design, complements pharmacokinetics. Basically, if you are taking the medicine, only then you will see how it goes through your body, how the mechanism is, how the effects are. So first the pharmacokinetics is done and then followed by the pharmacodynamics. So that complements to each other. Okay. So if you are taking one by one comparison of these two, pharmacokinetics and pharmacodynamics, so what it is that pharmacokinetics is the what the body does to the drug. Okay, so basically how body is digesting that drug, and the concentration over the time, how the concentration over the time in the body it is getting decreased because it is getting excrete from your body. And pharmacodynamics is the what the drug does to what the drug does to the body. So it is the mechanism of action and effect that drug plays into your body. So your body will obviously responds to that, and that response is known as pharmacodynamics. It's basically the process, whole process, not the response, but the whole process. So this is a drug receptor interaction. So basically, drug will go in your body. It will act to that particular targeted site, having a receptor, enzyme, or ion channel. So that will bind to that particular drug, and then they will do something mechanistically, and that will affect to your body. So this is how it is happening. We will be discussing these things in detail in the coming weeks. So now just I'm going to give you a basically overview of that particular thing. And pharmacodynamics effect over the concentration. So basically you will see the effect how many like doses you have taken, so you will see the effect throughout that. So this is how you can make a difference between pharmacokinetics and the pharmacodynamics. So is it clear to everyone? Okay, please, anyone has any doubt? Okay, so if it is clear to everyone and I'm moving forward, please, anyone has any doubt regarding anything on the slide, please ask me so that I can answer your questions. Or if I don't know anything, then also I will read it out and then I will answer your question. Okay, because my major is not in this particular thing but organic chemistry, so I'm not that much on that level, so I'm just also learning. So here, now going to the next question. So the main site of drug action is the liver, receptor, kidney and intestine. It is a very easy question, so can anyone answer it? Receptor. It's a like go question. So here the explanation is the drugs usually act by binding to receptor, obviously. So here, if you inject something in your body that is known as drug, then it will not directly go to your liver, directly go to your kidney, intestine and do something. It is not like that. It will go to your cell membranes, having a phospholipid layer, having a receptor, enzyme, ion channels attached to it, inside it. So then the drug will bind to one of the receptors, and then the action mechanism of action will start. It is not the rocket science that something is going in and it is attacking on everywhere and then it is responding it is not like that. So drug which is selective to a particular target is getting injected. It is going on to that particular selected target, and then the target having the receptor or anything else like enzymes and ion channels which will bind to that particular drug in such a way that you will cure that particular disease. So this is how the mechanism of action of that drug is. So drug first goes to your receptor. So receptors are a macromolecular proteins that is situated at the cell surface or inside the cell that recognize and bind specific ligands, drugs, hormones, or neurotransmitters and initiate a biological response. So basically, when it will bind to your drug, it will definitely going to initiate a response that is known as a biological response because it is happening in our body. So it is known as biological response. Okay. So these are the receptors. So is it clear to everyone? Okay. So now next question is, which property of a drug most strongly influences the protein binding? It is a molecular weight, lipophilicity, color or taste. What do you think? Lipophilicity. Right. So why lipophilicity? Because here they are asking which property of drug most strongly influence the protein binding. See, proteins are the basically the amino acids, right? So protein, your cell membrane have a protein layer, right? Amino acid layer or that lipophilic layer, which you can say is the lipophilic area is outward and the hydrophobic area is inward. Like in micelles, you will see. So this is how your protein binding is happening. So when your drug is going into your system and it is basically acting on the phospholipid layer, then the drug which is having a lipophilicity that will have a most influence on that particular protein binding because of the outer covering of that protein, that is a hydrophobic nature protein. So increased lipophilicity enhances the hydrophobic interaction with protein because the protein is having a layer which is outward, that is having a hydrophobic groups. So that is why the lipophilicity, which is also the lipophilicity means like the affinity to towards lipids, basically non-polar molecules, hydrophobic parts. So, if your drug has higher lipophilicity, then it will enhance the interaction between the outward layer of that particular cell membrane, which is of the protein, and hydrophobic interactions will get enhanced with proteins, which often increase the binding but sometimes reduce the free drug concentration. So basically, free drug concentration means that it is that much attached to your protein, the drug, that the free drug area is not left much. So that is why the lipophilicity is a strong ability of your drug to get influence with the protein binding. Okay. So basically, I think I'm having that particular phospholipid layer image in one of the slides. I will discuss it later. So here, is this clear to everyone, the question? Someone is joining now. Okay. Okay. So the answer is lipophilicity. So I am hoping that everyone is on the same page with me. Okay, now coming to the next question. So the primary step in drug action involves drug metabolism, drug excretion, drug target binding, and signal amplification. So the question is that first step which involves the drug action. So as long as you have taken a drug, so what is the first action which that drugs do in your body? What do you think? Okay. So drug target binding. Yes. So drug first of all bind to the particular biological target. And the target could be a receptor, a enzyme, ion channel, whatever it is. So these type of things are attached to your cell membrane and which is responsible for your drug action. So here the answer is drug target binding. So first of all, drug is getting attached to the binding target, and then we'll do everything which is written, but step by step. Okay. So is it clear? So as we have talked about the receptor, enzyme, ion channels, nucleic acids. So this is the brief picture of enzyme, receptor, and nucleic acid which are involved. So enzymes are the machinery of the cell and they catalyze the reaction. Receptors are the basically the compounds which responds to stimuli. Stimuli means that external activities which is happening in your body, it will respond towards it. And then the transmission of signals that is done by receptors. Nucleic acid, they store genetic information code. As we know that DNA, RNA are the nucleic acids. So here it is the vital for cellular functioning. Obviously it is very important for your cell to function. So these three things are important when you are going to think about the drug action and drug discovery. Today is the first session, so I am letting everyone in. But please make sure from next time onwards, please be on time. It will be saving your time as well as my time. And also less distractions, more chemistry we can learn. Okay? Okay. So these three are the things which is attached to your cell membranes. And of course the ion channels which are responsible, which I have not included here, but yes, there are some ion channels which are also responsible for your actions, for your absorptions, for your obviously ionic transport, sodium channel and potassium channel as we have already learned about in 12th class. So now coming to the next question, that is arrange the following non-covalent interaction in decreasing order of bond strength. Because when you are talking about the phospholipid bilayer, basically the cell membrane and the drug acting towards it, so you will see that drug and receptors and everything else, as we have talked, that enzymes or nucleic acid or ion channels, whatever it is. So when they bind with each other, so there are interactions, right? So in tutorial session also, professor has gone through that particular interaction one by one, which are van der Waal interaction, hydrogen bonding interaction, ionic interactions, and also that hydrophobic interaction, which we have already learned about in previous slide. So here, these interactions are all non-covalent interactions, right? So these are the non-covalent interaction, and also there is one more interaction which is the disulfide linkage, that is covalent, but that is found in the proteins. So you will be like learning about that in the coming slides. But here, what we are focusing on the non-covalent interaction, which are van der Waal, hydrogen bond, and ionic interactions. So now you have to arrange that interaction into a order, which is a decreasing order of the bond strength. So it is actually in the tutorial. So I have already just I have picked that question up in this session. Sir has already mentioned the bond strength in every interaction possible. So I hope whoever has seen already that particular video will be able to answer. So it is the van der Waal interaction, hydrogen bond or ionic hydrogen bond or van der Waal interaction, hydrogen bond, ionic and van der Waals, or dipole dipole, which is not in the basically dipole dipole interaction, ionic hydrogen bond. So, what do you think are the non-covalent interactions, and which is the order of that particular bond strength is correct? In decreasing order, I'm talking about. Huh? Option B. Okay. So, decreasing order of bond strength. Okay. Nice. So, option B is the correct answer. It is the ionic ionic interaction, which is ranging in between like I think 1 2 to 10 something. So that is the range of ionic interaction. Hydrogen bonding maybe 2 to 5, or van der Waal is less than 2 kilocal per mole, right? So this is how you can say that these are the interaction which are having the decreasing order of the bond strength. So the maximum energetically high and like basically the bond strength which is having the maximum strength is the ionic interaction, then the hydrogen bonding interaction, then the van der Waal interaction. So ionic interaction happens between the cationic and ionic charged species. Basically they will interact with each other. Hydrogen bond will happen between the heteroatom oxygen hydrogen, these kind of systems. Then van der Waal interaction is happening between the two non-polar species. Basically two alkyl groups is showing you the van der Waal interaction. So these are the different interaction. I hope you have already learned about it. So this question is dedicated to that particular thing, that you should know that how these interactions are arranged in the bond strength. Okay. Then going to the next question, but first of all, I will admit someone wants to in. I don't know why I'm keep getting those noises. Okay. Okay. So the question is, the basic structural framework of the cell membrane is formed by. This is a very basic question which I have already answered many times. So a cholesterol monolayer, protein bilayer, phospholipid bilayer, or glycoprotein matrix. What do you think? Yes, phospholipid bilayer. So this is the cell membrane are primarily composed of the phospholipid bilayer with hydrophilic heads and hydrophobic tails. Okay, so the outer parts of that particular membrane is having the hydrophilic groups, and the inner part is having the hydrophobic groups. So yes, here it is. So this is a phospholipid layer. This is the membrane cell, extracellular fluid which is having, and here which is this spherical systems are the hydrophilic polar head groups, and this long tail is the non-polar fatty acid chain that is the hydrophobic in nature. Okay, so this is how it is arranged. So I hope it is clear to all of you that how the cell membrane having a phospholipid layer, and there is a channel which is attached here like this. You can place here a receptor. Basically, this kind of receptor is here. If I will draw it correctly. So this is how the receptor is embedded in between this particular cell membrane. Okay. So this is how the receptor will look like. It is embedded to the cell membrane in different area, and then the receptor, then the drug molecule will add onto the receptor, and it will uh, do the things. So here the cytoplasm inside the cell membrane. So now coming to the next question based on receptors. So receptors are best described as lipids that store drugs, proteins that catalyze reactions, protein that bind ligands, initiate a response, or carbohydrates involved in signaling. What do you think is receptors are? Option C. Yes. So option C is receptors are protein molecule that selectively bind ligands, drug, hormones, and trigger a biological response. As I have already talked about the receptors, that it will bind to your drug molecule and it will trigger a biological response in the uh, whatever the action of response is there. So it will trigger a response in your body, and it will then go to your target thing. So next question is, enzymes differ from receptors mainly because enzymes are not proteins, do not bind ligands, catalyze chemical reactions, and are located only in membranes. So what do you think? Because enzymes are different from receptors. Sorry. Yeah, yeah, option C. Because enzyme catalyze the biochemical reaction, whereas receptors primarily initiate signaling. So this is how you will see that. enzymes catalyze the biochemical reaction, but whereas the receptor primarily initiate the signaling. So these two things are different from each other, and it will play a different role in your biological system to initiate a response or to basically do their jobs. Okay. Then proteins are primarily composed of fatty acids, nucleotides, amino acids, or monosaccharides. Amino acids. Yeah. So proteins are polymers of amino acids linked by peptide bonds. So I am actually keeping it very simple right now because the level of the first week is here and we have to build up on that. So you should be knowing the particular like basic chemistry from which you can develop that particular 12-week course, so that you can be having all the knowledge from the basic, from the start to the very end. So we are keeping it simple this time. Okay. So coming to the next question, amino acids and proteins are linked together by hydrogen bonds, ionic bonds, peptide bonds, or disulfide bonds. What do you think? Peptide bonds, right? So peptide bonds is a amide linkage. It is a amide linkage between carboxyl group of one amino acid and the amino group of another amino acid. Okay, so there is basically one amino acid here and there is another amino acid. So basically the hydroxy carboxyl group of this amino acid will bind with the amine group here. The basically you will know that nitrogen having the lone pair of electron will attack onto this carbonyl, and this double bond character will lose and O minus will get generated. After that, this O minus will then come down and it will leave out this particular H2O as a byproduct, and you will get a nitrogen carbon bond generated, that is known as a amide bond. So basically this amide linkage has a specific name because it is now having every amino acid is linked towards this, so it is having a specific linkage that is known as peptide bond. This carbon nitrogen bond highlighted as green is a peptide bond. It's some basic amide linkage, but it is known as peptide bond. Okay. So when you are forming two amino acid, basically you are clubbing two amino acid together, you will form a dipeptide. You are clubbing three amino acid together, you are forming the tripeptide, and that is how the chain will grow, and you will get a polypeptide or some things like that. Okay. So this is a condensation reaction in a short way. Okay. So now coming to the next question, the secondary structure of a protein is mainly stabilized by disulfide bonds, ionic interaction, hydrogen bonds, and hydrophobic interactions. So what do you think? We are talking about here the secondary structure of protein. Disulfide bonds. Okay. Any other thing? Any other answer? So here the explanation is hydrogen bond, guys. Why? Because you're talking about the protein. So we have already learned about it that protein has three structures. Basically, the primary structure, that is the linear chain one. Then you will go through the secondary structure having a alpha helix and beta sheet. Then you are going to that tertiary structure and then quaternary structure. So these four structures are there of protein, and it is actually folded way. So linear one is the primary one, secondary one is the pi stacking or something like that is happening. After that, there is a tertiary structure, and the quaternary one is the complex one. Basically, the having the various units attached to each other. So this is how it will look. The alpha helix, this system, it is the structurally it is this, and the beta pleated sheet structurally it is following this. So when you are talking about helix, so these helix having the hydrogen bonds, disulfide linkages are not possible here because the sulfur sulfur bonds, sulfur sulfur groups are not there, right? You are dealing with amine hydroxyl groups, sorry, carboxyl groups and amines. As we have already seen here, two peptides link two amino acid linked together to form a dipeptide is forming a amide linkage rather than a disulfide linkage. So that is why it is not possible to having the sulfur in these moiety. So there is no option of having that disulfide linkage. Instead, it is forming a hydrogen bonding with this helical form. So the carboxyl group of one helix is forming the hydrogen bond with between this amine. So this is how they are arranged as the alpha helix. And in the beta pleated sheets, what happens that you, if you are having a sheet like structure, so the amine group now will form a bond with the next sheet down there. So basically a stacking is there. So they will form a hydrogen bonding like this. So as alpha helix is a spiral linkage and the beta pleated sheet is a more or like a stacking position. So this is how they are forming the hydrogen bonds with each other, stabilizing basically the secondary protein of the secondary structure of the protein. Okay, is it clear that how hydrogen bonds are there in the secondary structure of the protein? Okay, so now going to the next question. This is the disulfide bonds in proteins are formed between two glycine residue, two cysteine residue, two lysine residue, or two serine residue. So for that, you need to know the structure of that particular amino acids. So what do you think? Option B. So it is between the two cysteine residues. So to answer this particular question, you should be knowing the structure, exact structure of these four amino acid: glycine, cysteine, lysine, and serine. So if you find the absolute structure, you will see that there is a disulfide linkage which is happening in these two molecules, that is cysteine. So how it is happening that when you club these two molecule together, so after oxidation it will form a sulfur sulfur bond that is known as disulfide bond. So here, this is how, this is not possible when you go to glycine, lysine, or serine. So this is not possible, this type of disulfide linkage is not possible in that case. Okay, is it clear to everyone? Okay. So today we have already discussed plenty of questions. So till now, if anyone has any doubt, please raise a hand. Please ask me anything which you have not understood in these particular slides. Any question? Yeah, actually, I am placing these slides into the public sheet, which coordinators have provided. So I will be uploading these slides on the particular public sheet, and you will, I think, can be, it could be accessible. I think because they are actually providing a sheet, so why would they provide that if they are not getting you to get accessible to it? So I think you will get these slides onto particular page. You will just have to ask the coordinator, and then I think you will get these slides. So, anything else? Yeah, it should be on every week, like on Saturday at 5 p.m. So, it is continuing till 12 weeks. So, it is the first week, so 11 weeks are left. So, every week we will be meeting, same time, same day, every week. Okay. Sorry, I can't hear you. Actually, we are two TAs for the particular course. So I think some other TA is there. So she will be taking your class from Monday. Yeah. So two TAs, two classes. Okay. Anything else? Okay. If nothing is there, then we will be just see, for your understanding of the medicinal chemistry, this particular table is very important, as I have already discussed, and I have already make so many questions out of it. So, if you are thinking of making a drug, then you have to test in a lab, then you have to go for the early clinical study, then the late clinical study, and then to the market. So, these are the four stages basically. So, for these stages to happen, you will have to find a lead molecule first, then you will test it on an animal test the toxicity, to test what is the uh, basically the preferred dosage or the toxicity which is there for the particular drug, and then you will test it on a small group of people, and for the small doses to get a hint that how it is behaving in human body, how the toxicity is there, how the mechanism of action is there, how it is how your body is actually responding towards it, and then it will actually take 14 to 18 months. So it is a very tedious process, a very long process to handle, and then you will have severe challenges of having a phase 1 to phase 3 trial. So a group of people is going to have your drug, and a group of people will be having different you know responses toward your drug, and you have to actually think about it, that from a individual perspective, that how you can overcome that safety hazard, how you can overcome that effect of every single individual, and then you can basically eliminate all the hazardous possibility, all the things, and then you will move forward to the phase 2, which is basically the efficacy. As I have already talked about, that efficacy is nothing but the efficiency of that particular drug. So how potent is that drug in your targeted area? So how that particular drug is basically giving a higher response toward that targeted area, and then you will see that how optimal dosage can be, like you can think of that optimal dose, because first the efficacy is sorted out, then you will see that you know that now this drug is that much potent to that particular disease or that particular target molecule, and then you will see that how you will come up with the optimal dosage, you will come up with a minimal dosage, so that your body is not uh, basically responding hazardously towards that particular dose, right? So everyone has different responses, so that is why this is a very important clinical trial of phase 2, that will actually suggest you that how you will play with that particular dose, and also how you can uh, think of using that particular dose to majority of people having 50 to 300. So this thing will take your 2 years straight away, because it will take a very much tedious process, or a very much, because everyone is not that much capable of having doses of the particular medicine, because uh, as you can see that when you are taking one pill, you are having something, if you are getting treated, but you will think that maybe I will take two, then I will basically think that this particular dosage is well enough to be treated on that disease, but you are exceeding that dose, right? You are exceeding that optimal limit, you are exceeding that hazardous limit. So if you are taking too many medicine, too many pills, you will end up having that particular adverse effect on your body. And that also, doctors will say that please don't take too much of medicine, please don't take two pills together, please don't maintain a distance, or maintain a hour distance between one doses and two doses. So that is why they have told you these things, because the adverse effect, the things which may be very light for your thinking, will not go in the biological system. In the biological system, it will have hazardous effects. So this basic medicinal chemistry course is dedicated to that things, that you will take into the mind, you will take into your home, that these things is not like you start a day, you will discover something, then you get it manufactured. It is not like that. Okay, it takes a tedious process, it takes so many people to discover, to go through that clinical stages, late clinical stages, and then the market unit is a all different story, because there also marketing, the drug should be cheap, right? It should be accessible by a large group of people. It is not limited to some group of people, so you have to maintain that thing also. So every stage have a different setup, every stage have different units, different people working together. So the medicinal chemistry basics will be going into this particular chart. So I will be more than happy if you focus on this particular chart, and if you just think about it, learn about it, that how the things are happening in every stage. Okay. So please do read the tutorials before coming to this particular do read the videos before coming to the tutorials, because that will help me and that will help you to answer the questions. Today I will set the bar very low because this is the first week and everyone is not prepared mentally, as I am also not prepared mentally. So I will be thinking that everyone is like me. So guys, today we will be having a basically we had a basic knowledge test, and then we will move forward very slowly so that everyone can get a pace of it, everyone can get understanding of the medicinal chemistry course. It is a bit tedious course, but it is actually implementing in your daily life, so it will be interesting in that aspect also. Okay, so I hope everything is clear to everyone. So now if anyone has any doubt regarding the meetings, regarding the tutorials, regarding the presentation, anything, you can ask anything right now, and we will be meeting on the next Saturday at the similar time, 5. And please make sure to join on time because it actually takes some amount of concentration to get you joined and then coming back to the slides and then again starting from the scratch. Okay. So okay, 16 people are there. I have actually joined 23. अच्छा तो मैं बोर कर रही थी. Okay. Okay. If you say so. So I hope you guys are having a good time with me. So just end this lecture today. And if you have any question right now, please ask me right now so that I can end this session and we will be meeting next Saturday. Any questions for now? Okay. I suppose that there is no question for today. So I will be ending this meeting today and hope to see you soon on the next Saturday with the great amount of numbers. Okay. So thank you so much for attending. Thank you. Thank you. Thank you.